Thank you. Okay, so we're back to contributed talks. And our first speaker for the rest of the session is going to talk about vectorized error signals in cortical dendrites during a brain-computer interface task. And the talk is presented by Valerio Francioni from the Harnett lab at MIT.
So good afternoon. First, I want to thank the organizers for this amazing conference. I've had a great time so far, and for giving me the opportunity to present my work. So let's dive into it. In the brain, learning happens through changes in synaptic strength. And for a long time, the dominant model for learning in the brain has been reinforcement learning, which in its most Pavlovian sense is a form of scalar learning in which when the animal does something correctly, all the synapses which were recently active are reinforced through a global teaching signal. However, scalar learning has limits, and if we train machine based on scalar learning alone, learning is slow and often ineffective. And so a second algorithm to train artificial network is called backpropagation. In backpropagation, error signals are vectorized and targeted to specific synapses, providing a much more precise teaching signal. Backpropagation has been at the core of the recent machine learning revolution, and its efficiency prompts the question as to whether error vectorization is just a convenient machine learning trick or an approach which biological brains also employ during learning. However, historically, backpropagation has been considered biologically implausible. Part of the reason has to do with the fact that in order to do backpropagation, neural networks, whether they're artificial or biological, they need to independently process feed-forward sensory information from feedback teaching signal, as these two streams of inputs convey qualitatively different type of information. In machine learning, neurons are represented as point neurons. And since point neurons cannot effectively separate feedback and feed-forward and feedback signals, this separation happens temporally. However, temporal separation seems implausible in the brain, which receives a constant flow of feed-forward sensory input. Some recent theoretical papers, however, suggest that the brain could separate feed-forward and feedback inputs spatially onto the soma and dendrites, respectively, and approximate backpropagation during learning. This spatial segregation hypothesis maps nicely to functional and anatomical data that show the individual pyramidal neurons receive bottom-up inputs close to the soma and top-down inputs onto the distal part of their dendrites. And despite its importance, this hypothesis remains untested. There are two main practical problems with testing this hypothesis. The first one is empirical. If soma and dendrites process and receive different type of inputs, we would expect their activity to be somewhat different. And despite we know that dendrites have the biophysical apparatus to do independent computation, in vivo observations show that somatic and dendritic activity are highly correlated under GCAMP regime. The second issue is technical. How do we define an error at the single cell level? In artificial neural network, the cost function, which is the causal mapping between single neuron activity and performance, is known. In the brain, however, this is different. And this is because it's unknown whether brains represent the cost function explicitly. And even if they do, experimenters would be typically blind to it. And so to solve this problem, we designed a brain-computer interface-based learning task. And here we image the proximal trunk as a proxy for somatic activity of layer 5 pyramidal neurons in the retrosplenial cortex. From all the neurons that we had in our field of view, on average 250, we then selected 8 to 10 neurons randomly, and we split them into two arbitrary populations. You can see them in here in red, P+, and in blue, P- neurons. Activity in the P+ neurons would rotate a stimulus clockwise towards a target 90-degree angle, which is when the animal would receive a reward. And on the other hand, activity in the P- neurons would rotate the stimulus in the opposite direction, away from the target. So mice had to selectively activate P+ or inhibit P- neurons in order to receive a reward. Here's what the data looks like. In red, you can see activity from 4 P+ neurons. In blue, from 4 P- neurons. Their activity in each subpopulation is averaged together, and the mean activity from these two subpopulations is subtracted from one another. And this subtracted signal is then used to drive the rotation of a visual stimulus with a target angle which is known at any given time. So as you can see from this cartoon, this allows us to unambiguously define a moment-to-moment error signal as the distance between our current and the target activity at any given point. Over time, mice increase both in accuracy, which is a fraction of rewarded trials, and in the speed of performance. And this increase in performance is driven by the differential regulation in the activity of P plus and P minus neurons over days. So now we have a task that mice learn with an explicit cost function, and we can look at the dendrites. Empirically, despite the onset time of somatic and dendritic signals remained highly correlated, even under our experimental condition, we noticed that the relative amplitudes of GCAM signals changed. An ex vivo characterization of GCAM signals previously done in our lab demonstrated that once we correct for the number of somatic action potentials and dendritic spike duration, GCAM amplitude differences reflected current injected in the soma versus current injected in the apical dendrites, reflecting a spatial bias for inputs. So now we want to know whether relative changes in amplitude signals could be predicted using activity in the surrounding network of neurons. How did we approach this? First, we identified individual coincident somatodendritic events. You can see them here labeled 1 to 5. And for each of these events, we estimated somatic versus dendritic event amplitude and fitted this relationship using a best fit linear regression. For each of these events, we defined the residual as the component of dendritic activity independent of the soma. And next, we define positive residuals, so everything above this fitted regression has dendritically amplified, and everything below this best fit line has dendritically attenuated. For each of these events, then, we calculated the high-dimensional population vector of activity as the activity in all the surrounding neurons in the 2 seconds preceding event onset. In the bottom right graph, you can see the population vector associated with each of the events in this upper right panel, color-coded according to whether the event in the dendrite was amplified or attenuated. And finally, we used a linear SVM to try to predict attenuation and amplification using activity in this surrounding network of neurons. We found that we could predict amplification and attenuation with over 60% accuracy. And interestingly, by estimating the distance from this maximally separating upper plane, we could also predict how much amplification or attenuation we would see in an individual somatodendritic event. Importantly, our approach completely decorrelates somatic activity from dendritic activity. And this is, yes, a nice control because it suggests that we're not just decoding some spillover information from the soma, but it also suggests that whatever information is encoded in the dendrites must be encoded de novo independently of the soma. And so next, we wanted to know whether we could use the somatodendritic residual at the population level, so for all the neurons that we had, to decode the task-related variables. And to do so, we estimated the residuals for rewarded trials in the 2 seconds preceding and following target reach from the animal. While in all the unrewarded trials, we looked at times 2 seconds before and following the end of a trial. Using these population vectors of somatodendritic residuals, we were able to decode rewarded versus unrewarded trials, demonstrating that individual neurons encode information about the network states that correspond to successful versus unsuccessful outcomes in their dendrites. Finally, we want to test our last hypothesis. Can the brains use dendrites for computing errors and for vectorized learning? To do this for P plus and P minus neurons, we evaluated the somatodendritic residuals at times in which the overall error increased versus times in which the overall error decreased. There were 3 possible hypotheses for this. The first one is that the error was not encoded in the dendrites at all. The second one is that the error would be scalar. So P plus, the prediction would be that P plus and P minus neurons would receive the same error signals. And the third hypothesis is that the error is vectorized, so that P plus and P minus neurons would receive different error signals according to their causal contribution to the task. Here you can see delta F over F traces in the soma and dendrites in black and orange, respectively, for a P plus neurons during epochs of error reduction and epochs of error increase. As you can see, the ratio between somatic and dendritic activity is higher during epochs of error decrease compared to epochs of error increase. In the P- neurons, on the other hand, this relationship is inverted, in line with the vectorization hypothesis. This inverted relationship is maintained at the population level, and so error decrease produces more amplification in P+ neurons compared to P- neurons, while the opposite is true for epochs of error increase. And so, in summary, we can use the BCI to map neuronal activity, behavior, and error, and study the basic properties of learning. We show that the somatodendritic coupling can be predicted from activity in the local network of neurons. The somatodendritic coupling contains de novo information about task-related variables that could be used as instructive signals. And the error signals were vectorized and selectively computed at the distal dendrites of pyramidal neurons. With this, I want to thank first Mark, who's an inspiring scientific mentor and a compassionate human being. Everyone in the Harnett lab for the supportive working environment they provide every day. It's a great place to work in. And more specifically, Vincent and Norma for their invaluable help with different aspects of this project. And finally, I want to thank you all for your attention today, and I'll be happy to take questions.
We have time for a couple of questions.
Hi, Valerio. Great talk. In your view, are the signals that are synchronous in the soma and dendrite, do you think they're being backpropagated? Do you think they're being forward propagated? And does that have any different interpretation for your model?
Sorry, could you repeat the first part?
How much of the synchronous signals do you think are backpropagated versus forward propagated?
Oh,
and does that have implications for your model?
Right. So the question is whether they come from the soma, whether they're received on dendrites. So it's really hard to tell because of the temporal dynamic of the GCAMP indicator. We see that the events, though, that are more dendritically amplified, they are at a shortened latency compared to the soma, so suggesting that they tend to arrive earlier compared to the ones that are dendritically attenuated. In general, I think that there's a broader issue about just thinking about whether the events in the dendrites come from the soma, whether the events in the soma come from the dendrites, which is related to the fact that once we get into these burst modes, and we know that all the events that we're seeing here are bursts because we characterized the GCAM previously, we know that somatic action potentials can actually activate calcium channels in the dendrites, and that in turn that leads to plateau calcium potentials that in turn activate the soma. And so we think that there's this like back and forth mechanism, and that is very feedback driven, let's say.
Balázs?
Thank you.
Do you image during the process of learning, and when do you see the maximal effect or difference in the dendritic?
Sorry,
Do you do imaging throughout the whole learning process, and when do you see the maximal difference? In naive animals or in expert animals?
Oh, yeah. So that's a great question. So we have started to look into that for when these teaching signals start to appear, and how do they relate with like the dynamics. The analysis is still preliminary, and so I don't have a full answer for you yet. But we see a lot of interesting instructive signals already from the very early days. This is not very surprising. There was a Neely paper that does similar experiments with in the Carmena lab with spikes, so with electrodes instead of imaging, and they show that mice can learn actually to perform the task even within the same day. So the learning is actually quite fast.
Okay, let's thank Valerio again.
Thank you very much.
So next we have Yuhan Liu, who is going to tell us about biological plausible backpropagation across arbitrary time spans via local neuromodulation.
Good afternoon, everyone. My name is Helena. I'm a PhD student in applied math at the University of Washington, advised by Professor Eric Shea-Brown, and I have also been collaborating a lot with the Allen Institute for Brain Science and Mila AI Institute here in Montreal. So in the study that I'm presenting today, we propose that local neuromodulators such as neuropeptides can help to approximate backpropagation through time. So guess what this is? It's a recurrent neural network. Something we've been hearing over and over again for the past few days. And the magic usually comes from the connection weights, which you can adjust to produce a desired input-output mapping, and how far your output deviates from the desired target for the task is usually quantified by this loss function E. And the question becomes, how do you adjust the weights so that you can produce desired input-output mapping for the task objective? So in artificial RNNs, this is a solved problem because one can just use backpropagation through time, or BPTT for short, which is the temporal analog of backprop. So what BPTT does is it computes the gradient of the loss with respect to the weights, and that informs how changing each weight impacts the loss. How changing each weight impact the loss will depend on many neurons in the network. So that will not only depend on neurons I and J immediate to the connection, but also because we are now dealing with a recurrent neural network, the activity will propagate to the rest of the network through recurrence. So changing WJI can also affect this blue neuron K that will impact the loss, and this red neuron L, and this pink neuron M, and so on. So in other words, updating just a single weight will have impact that propagate throughout the network. And this will require a vector feedback update WJI. I just want to mention that in the previous talk, you heard about vectorized error signal again, but the previous talk is more focusing on specificity to the receiver, whereas here when we talk about vector feedback, we're talking about each receiver receives many feedback signals. So here, just updating WJI, if you use BPTT, they will receive feedback from multiple sources from all of these neurons, informing how changing WJI impact their activity and hence the loss. So I mentioned this as a solved problem in artificial RNN, but our community is more interested in how can RNNs in the brain implement the vector feedback, which is the focus of my talk today. And as I'll explain more later, that it's now straightforward because we're dealing with recurrent neural network, which pose challenges now seen in feed-forward settings, as BPTT requires expensive information to be fed back across both space and time. It's unclear how neural circuits have the machinery to support that. So one proposal people propose is that what if we use a simpler feedback? So instead of having vector feedback from all these different neurons, what if we ignore contribution feedback from all of these color neurons and just get a simple scalar feedback as in the standard 3-factor rule? And we know that such scalar feedback can be delivered to each synapse because a well-studied candidate for that could be a neuromodulator dopamine, which usually comes outside of cortical network and can act in a top-down manner, informing how the cortical network did overall. These 3-factor rules actually perform pretty well, but there's still a performance gap from BPTT, so that's why we don't stop here and wonder if there are missing mechanisms that have yet to be accounted that could bring us closer to BPTT. In fact, there's this recent transcriptomic data from the Allen Institute. They uncovered that in addition to dopamine, there are many modulatory signals secreted within the cortical network, such as these local modulatory signals, such as neuropeptides. And these neuropeptides could potentially offer us additional feedback dimension for more informative feedback. So these local modulatory signals come in multiple types, and each of them form a separate feedback signal. Because the number of types is much less than the number of cells, the dimension of the feedback through such mechanism would be less than that of the cell-specific feedback in BPTT, but more than the scalar feedback in three-factor rule. And moreover, these local modulators, such as neuropeptide, can modulate synaptic plasticity just like dopamine. But what really sets them apart from dopamine is that their expression is ubiquitous within mammalian cortex. So what that would imply is that this endows nearly all neurons' ability to send modulatory signals to each other, and hence could potentially signal feedback locally within the network. So this motivates us to ask, what if we bring these local modulators into the equation? Can we arrive at a learning rule that comes in between BPTT and three-factor rule? And we'll later show that indeed that's the case. These local neuromodulators can help to implement a coarse-grained version of the feedback signal in BPTT. And we call our rule ModProp, which stands for modulatory propagation. So the main question is, how might local modulatory signals, such as neuropeptides, help to guide weight updates in RNNs? And a spoiler for our story is that we show local modulatory signals, such as neuropeptides, could help to implement a coarse-grained version of the vector feedback in BPTT. And as a spoiler for our results, which I'll go more in detail later, we measure the performance of different learning rules using the learning curve, which is a loss reduction of a training iteration. So this is one for BPTT, for three-factor rule, and as expected, our rule performs in between them. I'll explain this plot again later. Okay, so before I talk more about our rule ModProp, let's first come back to this RN diagram I showed at the beginning. And recall I said that BPTT tries to figure out how changing each weight would impact the loss. And how changing each weight would not only directly impact the loss. What I mean by direct impact means influence on the loss through neurons immediate to the synaptic connection, but also indirectly influence the loss through many other neurons in the network due to recurrence. So for a learning rule to be biologically plausible, all the variables involved in the weight update must be physically available to that synapse. But here, if we were to use BPTT, it would involve communicating this indirect impact term that requires sending activity of nearly the entire network to every synapse. And it's unclear how neural circuits could support such expensive operation. So some recent well-known studies by Bellick et al. And Murray propose how neural circuits can partially implement BPTT. And what they did is they focused just on the impact of weight change on loss through neurons immediate to the connection. That's neurons, presynaptic neuron I and postsynaptic neuron J for weight JI. And by doing that, Bellick et al. Decomposed the weight update into two components. The first captures how cell J's firing influence E, and the second captures how WJI influence cell J. And they were able to map the first onto top-down learning signals such as dopamine that can steer plasticity, and the latter onto eligibility trace that keeps a fading memory of coincidental pre- and postsynaptic neuron activation. So building on their great work, we wonder if we can also assign putative identities of the indirect impact term to biological signals if we bring local modulatory signals into the equation. So our first step is to decompose this into multiple terms, each capturing which neuron is WJI impacting. And then before we try to map it to local modulatory signal, as a reminder, it should obey the two properties I introduced earlier. So first, it can modulate synaptic plasticity just like dopamine, so which means similar to how top-down learning signal can be combined with eligibility trace, local modulatory signals should be able to do that too. And second, their expression is ubiquitous and cell type specific. So it turns out we can incorporate these properties into these terms with some approximations. So we follow similar steps as in Valerio and again arrive at the modulator signals combined with eligibility trace. So recall that each neuron secretes a local modulator signal, they reflect how that neuron's firing impact the loss. And then once these local modulator signals are secreted, their effect on the receiving target is captured by some cell type specific modulatory weight matrix B, because as I mentioned, the expression of these neuropeptides, precursors, and receptors are cell type specific. And as shown on the top right of the slide, the entries of B is only specific to every sender receiver type instead of every sender receiver cell. So this results in a coarse-grained low rank matrix. So this is why the feedback delivered through such mechanism is lower rank and less complex than the full cell-specific vector feedback in BPTT. And moreover, if we assume some correspondence between neighbor order and type and the timing they take effect, this allows us to group these additional terms into a single convolution form. So instead of, so like three-factor rule, we here also combine eligibility trace with modulator signals, but in three-factor rule, they are combined through multiplication. Here, they can be combined through convolution. And another highlight of our rule mod prop is that feedback can be delivered through cell type specific and coarse-grained local modulator signals. So I mentioned that our rule is a coarse-grained version of BPTT, and specifically two approximations were made in that process. So as I mentioned, the feedback modulator weights are cell type specific, and here we use two types, which corresponds to 4 different values. And another thing to note is that we had to make some additional approximations for the modulator weights to be timing variant. So how well does this coarse graining work? We tested on the simple pattern generation task, but also tested a few other tasks in the paper. So as I mentioned, we measure the performance using a learning curve, that is the loss reduction over training iterations. And this is the one for BPTT, our gold standard. It uses full cell-specific vector feedback. But as I mentioned, the bioplausibility of BPTT is questionable. And then so here's the one for three-factor rule. It performs slightly worse because it leads to less loss reduction. And that's expected because three-factor rule uses less informative feedback than BPTT. But the trade-off is that it's biologically plausible, which is important if we want to build a valid model to study neural circuits. And as expected, our rule performs in between them, thereby further advancing what bioplausible rules can do. So in conclusion, we propose that local neuromodulators such as neuropeptides can help to implement a coarse-grained version of the vector feedback in BPTT, thereby leading to a rule that performs between BPTT and three-factor rule. And I should also mention there's still ongoing work. There are several limitations that will need to be addressed in the future, such as we only tested our rule on a limited set of tasks and architecture settings. And also, I briefly mentioned we only use 2 modulatory types, but transcriptomic data hints 20 plus types. So the role of such cell type diversity is also left for future investigations. And lastly, another future avenue could involve studying the implication of our rule for neuromorphic computing. More discussions on limitations and future work can be found in our paper. Okay, so lastly, I would just like to thank all my mentors for helping me out in hundreds of ways. And we would also like to thank all members of the Shea-Brown, Fairholt, Walker, and Ledra groups. And thank the funding sources, thank the organizer for organizing such an amazing conference, and thank you all for your attention.
We have time for one quick question. Go ahead.
Thank you. My question is related with the neuromorphic side of your work. So in the EPROP, the Guillaume Spalek's work, depending on the neuron type, you can have per neuron or per synaptic eligibility traces, right? I was wondering the eligibility traces you use, whether they can be calculated per neuron or is it per synaptic?
So if I understood your question correctly, you're asking about the dimension of eligibility trace. So here, eligibility trace is specific to every synapse.
Okay,
Is that your question?
So, can the next speaker please come to the podium? We're not seeing you. Okay, so while she's setting up, I can ask a quick question. So I'm a little bit confused about how this works biophysically. So are these neuropeptides substances that individual neurons release in an activity-dependent way, and those diffuse to the rest of the neurons in the network?
Mm-hmm. Yes.
And everybody's supposed to do this at the same time? Aren't you kind of in a soup of neuropeptides that kind of flat over time and over neurons?
That's a really good question.
Where's the information there?
So once they're sent, right, how they are detected, how they take effect, it could depend on multiple things. That would depend on the sender and receiver types. That's where the cell type specific modulated ways come in. So some neuropeptides that don't there are some neurons. So it's cell type specific. So some neurons may not express a receptor that listens to certain neuropeptides, and if that's the case, these won't detect some neuropeptides. Yeah, thank you.
Thank you. Okay. Let's thank Helena again. So we're moving to Sol Markman, who's going to talk about the geometry of sensory and cognitive representations and how it implies the population's causal impact on choice.
Thank you. Hello, everyone. It's an honor to be up here presenting this work to you. Today, I'll be talking about how the geometry of sensory and cognitive representations relates to a population's impact on behavioral choices. And before I start, I want to quickly acknowledge Doug Ruff, who did all the monkey experiments I will talk about today, and Jason Kim, our collaborator, and of course, Marlene Cohen, my wonderful PI. So real-life decisions are based on multiple sources of information. Imagine that you're having a movie night and you're sitting down to choose a movie to stream. You might consider what genre you're in the mood for, who are the main leads, what reviewers have to say, or a friend's recommendation. And as you're making this decision, you're probably combining many of these factors to arrive at one final movie choice. And lots of recent work has shown that these multiple information sources are represented in a surprising number of brain areas at once. And the fact that everything seems to be everywhere is puzzling because different brain areas are known to play distinct roles in decision computations. For example, in a motion estimation task, we think that visual areas mostly represent the sensory information, while frontal and parietal areas compute the decision and enact the choice. We investigated similarities and differences between neural populations in these two stages of decision-making by training monkeys to make choices based on emotion stimulus and reward information. And we chose to record from areas MT and DLPFC because of the large body of work that places them well within these roles. And in the years since these initial experiments, we as a field have started thinking about concepts like the geometry of representations on the population level. So in this work, we found that the formatting of task variables relative to each other may be an important clue to different populations' causal role in computing decisions. And we hope that what we learned by studying the simple system will help guide future studies of less understood brain areas and behaviors. So in the base version of this task, a monkey is presented with a random dot motion stimulus, which can be moving in any direction in the 2D plane. The monkey reports the perceived motion direction by making an eye movement to a location on the ring, and they are rewarded according to the accuracy of their perceptual judgment. Simple enough. But in the full test, we've added an additional source of information that the monkey must consider, a reward bias that modulates the reward amount, where the closer you are to this pink region, the higher the amount of juice that you get. And if the monkey only considers the reward bias and ignores the stimulus, they would saccade to the center of the pink region. But the total amount of reward is a function of the motion direction and the reward bias in this test. So to maximize rewards, the monkey's choice should actually lie between these two locations. And indeed, we see that the monkeys are smart and after training have learned how to make choices based on both the motion direction and the reward bias. So if the monkey was only reporting the motion direction, then their choices would lie along unity. But we see that the choices are biased towards the high reward region at 60 degrees and away from the low reward region at 240 degrees. And if we now switch the reward bias, where the pink region is at 240 degrees, you see the opposite effect. Now there's a repelling around 60 and a bias towards 240. And while the monkeys were doing this task, we recorded from areas MT and DLPFC using 24 or 32 Plexon B or S probes. And both motion and reward information are decodable from each area. However, as you'll see in the following illustrations, these sources of information are formatted differently. In this first plot, I'm showing the MT population activity from an example session, plotted in 2 dimensions that contain the most information about motion direction. And filled circles indicate one reward bias condition, while open circles indicate the opposite condition. And you can see that in these motion dimensions, you get very nice rings organized by motion direction, but very little separation of the 2 reward bias conditions. But now if we find the dimension that best distinguishes between the 2 reward conditions, we see that MT does indeed contain information about the reward bias, but just in an orthogonal dimension. So now if we do the same analysis for DLPFC, we see that the 2 reward bias conditions are already separated in the motion plane. And likewise, the reward dimension also contains information about motion direction. As you can see, this ring here is also oriented with variance in the z-axis. So unlike in MT, motion and reward information are encoded in the same dimensions. And these example plots are just an illustration of this formatting difference, which we've also tested more rigorously with decoding analyses. But for the sake of time, I won't talk about that today, and you can ask me after. So to better understand the implications of different representation geometries, we developed, of course, RNN models that replicate features of the low-dimensional population structures, specifically the formatting of motion and reward information. So both models receive the reward bias and motion direction as inputs, but the MT-like model is trained to produce separable outputs, while the DLPFC-like model combines them to output the appropriate reward function, which is proportional to the reward available at each location on the ring. So here's what those output functions look like when the motion direction is at 0 degrees and the reward bias is at 90 degrees. After training, the DLPFC model outputs represent the expected reward at each location. So we can read out the model's choices as a direction represented by the output unit with the highest activity, which in this case should be 30 degrees. And so as you can see, the model reproduces the monkey's behavior pretty well. Here I'm showing you the 2 reward conditions of in the same plot that I showed earlier, but the model is trained on 36 reward conditions spanning the whole ring. And as we intended, when we look at the recurrent layer activity, we see that in the MT model, motion and reward variables are represented in orthogonal dimensions, while in the DLPFC-like model, they're represented in the same dimensions. So you can see a bit of separation of the 2 reward bias conditions already in the motion plane, and that the motion and reward dimensions contain the activity in the same plane in the 3D plot. And so here now I'm plotting all of the model's fixed points and all 2,600 learned trial conditions in the first 3 PCs. And the MT model can be understood by the previous illustrations, while the DLPFC model is much higher dimensional. And I could spend all day showing you these pretty plots, but what we really want to know is how these distinct geometries, including the formatting of task variables that I showed you in the data and the model, relate to differential contributions to behavioral choices. So how can we probe this? One way is to perturb the network's activity, for example, by stimulating units with different tuning preferences, and seeing how this changes choices. So in the RNNs, we model stimulation by adding an untrained input to single units in the recurrent layer during the stimulus presentation period. And so here's an example of what happens in the MT model, where we stimulated a unit with a preferred direction of 100 degrees on trials where the motion direction input was 0 degrees. And the resulting choices are shifted such that they lie between the motion direction and the stimulated direction. And we call this a vector averaging effect because the choices are a weighted average of the motion input and the stimulation input. And so increasing or decreasing the magnitude of that stimulation strength would smoothly shift choices more or less towards the red line. In the DLPFC-like model, we see something very different. Either the stimulation completely determines the chosen direction on a trial, or it has virtually no effect, and the model still outputs the usual choices, which are denoted by the green X. And so in this model, there's a competition between different choice directions, leading to a winner-take-all effect. So now the moment of truth, where we see whether the model's predictions hold in the neural data. And indeed, we see that with microstimulation in MT, we get a vector averaging effect. On the right is data from an example condition from one session, and on the left is the model's predictions for that same condition. And in DLPFC, we see that microstimulation leads to a winner-take-all effect on the choices. Either the stimulation has little to no effect, and the monkey's choices remain unchanged near this green dashed line, or the stimulation has an overwhelming effect, and the monkey chooses near the stimulated direction. And so those were just some especially nice example sessions, but here I'm showing that this holds even across many stimulated sites and motion directions. So here I'm showing more than 1,100 trials where the different stimulated directions are aligned at 0 degrees, and I've picked out trials where the motion direction is 100 degrees counterclockwise from that location. And you can see that the MT distribution is clearly unimodal, with choices shifted towards the stimulated direction, as shown by this. The mean is shown by this black dashed line with the orange marker. And in DLPFC, the distribution is bimodal, with peaks at the unperturbed choice direction, the green X, and at the stimulated direction, shown by the red line. And we've also done statistical tests to confirm that these distributions are unimodal and bimodal, respectively. So to summarize, I've showed you that motion and reward information were decodable from both MT and DLPFC. However, they were formatted differently in the two areas, consistent with different roles in decision-making. In MT, we saw separable representations of sensory and cognitive task variables and smooth integration of incoming information, so the vector averaging effect of microstimulation. While in DLPFC, we saw a combined representation of task variables that inform a decision, with winner-take-all competition between potential choices. Together, these results suggest that representation geometries are an important indicator of a population's causal role in behavior. And so I talked mostly about the formatting of task-relevant variables, but we are also very interested in how attractor dynamics, the dimensionality of activity, and control theory can help explain how perturbations affect behavior in the system. And we believe that understanding the relationship between these concepts will help us understand the function of less understood neural populations in more complex behaviors. And so with that, thank you all for listening. Thank you to the amazing Cohen Lab, now at UChicago, and to our collaborators, especially Jason Kim, who's at Cornell.
We have time for a couple of quick questions, America.
Hey, Sol, nice work. Quick question. I'm sure you know Jamie Nichols studied microstimulation in MT.
Yes.
So that result suggests that the vector averaging happens when the difference between the angles is on the order of 90 or less, and beyond that it actually becomes a winner take all. How does that work play out in this experiment?
Yeah, we actually, so we didn't see such a clean winner take all effect in MT, and we think it's because of the reward bias, but we do think that that result is definitely true, and it also holds in our models that once you get beyond about 130 degrees, you start to see winner take all. And so you can think of that as just if you have two Gaussians with that are pretty wide, and you separate them enough, you start to get two peaks rather than one in the center. So that's how I think about it.
But that makes sense in terms of your interpretation of the geometries?
Yeah, I think it does, because Yeah, I think it is consistent because at least just empirically, we've seen in the models that you can have this geometry with a winner-take-all effect when the directions are different enough. Thank you.
That was awesome. Thank you so much. I have one question. I know that there's like not a lot of choice probability in monkey MT, but I was wondering if it's possible that you have enough neurons that are response coding to check whether the reward information like might be parallel to the response coding as opposed to like, I don't know. Yeah, is it possible that that's checkable?
Yeah, that's an interesting thought. I think it's a little hard to tell because the response directions are so similar to the actual stimulus direction.
Okay.
But yeah, it is an interesting direction to differentiate between the two.
Cool, thanks.
Thank you.
Let's thank Sol again. Connection. Had to have one of these. Going too well.
Hey, sorry.
It
It was great moments ago. So I unplug it and plug it back in.
That's what we're gonna try.
It did take a moment last time. Yeah. Seems promising. Okay. Apologies.
Again?
Yep.
A
I'll
different
pull
port.
this out. Yeah.
I think
sure.
that's fine. Sorry about that.
Oh, okay. Did you see it for a moment?
Change it up to.
Yeah, I'm gonna change it up.
So that and this one, if you don't worry about it.
I can just click the keys.
I mean, the clicker, this has a,
If it works. Yeah, exactly. It looks like it's not really the adapter.
no, it's not the stuff. The adapter or the computer. Because
But it literally was working just.
yeah.
You
When
want me
he-
to re something? I mean, I don't know what else. Or something?
Yeah.
Okay. Apologies.
Yeah, sorry everyone. It was bound to happen on this very last talk. It's in the spirit, exactly.
Let's turn it off and turn it back on again. Oh boy. Good stuff.
Yeah, no, it was on the screen just a second ago.
Yeah.
Literally just when you moved it. There is something physically not right about
Is
this
this,
box.
is it possible to actually pull the power in into that thing? Okay, we have another map. We have another computer. Okay, fine. I was gonna say, I can access it.
There we go. Okay.
Okay,
It just came up there a second ago.
Oh.
Yeah, okay, here we go. So far, so good.
okay. Oh.
Oh no, and is it online, the talk?
Yeah, I have a PDF which I saved, but it's pretty rough. Like that's like real emergency.
Okay.
Because it will, yeah. Oh, that's painful.
I can create a Wi-Fi hotspot.
Oh my God. That's crazy. Okay.
Okay, here we go.
Okay.
Excellent. So, with no further ado,
Okay.
Kimmy.
Hey, sorry. Many apologies. It's okay. Okay. We did test this, but you know, okay. I'm gonna get moving because I'm way behind. My name is Tim. I'm gonna talk to you about BCI and gradient-based learning today. It's great that someone's already talked a bit about BCI earlier, so some of it's loaded in our head. I want to take a moment right up front and talk a little bit for a moment and talk about who did a huge amount of the work here. This was Peter Humphreys. There were other people involved, but he really drove this work. He's a really talented guy in my group. Reach out to him if you're curious. Okay, I want to start for a moment and just like really zoomed out and ask this question about how should we understand complex animal brains? And in some other formats and some other places, I've argued that we don't really know how to understand complex network circuits that do things like recognize objects, play chess or Go, but we readily understand how to build them now. And I think that in computational neuroscience, my take on this, I've argued this a bit with Blake Richards and Conrad Korting, that we should focus instead on these things: network architectures, objective functions, learning rules, and the data that goes into generating the behaviors that we see, the complex behaviors that we see that we're interested in, rather than trying to understand how networks compute what they do, which I think we can't even solve in silico. Anyway, to that end, I got really interested in BCI. It seems like a super interesting place to go and study learning rules in particular. And why is that? Because you have animals that are hooked up in a learning situation where they're going to improve. They're definitely running some kind of a learning rule. And you have a lot of experimental control as an experimenter. You're the one saying, hey, here's how the neural activity is going to feed forward into a BCI mapping. And here's how I'm going to reward the animal based on what it accomplishes in the world. So I'm going to talk a lot in this talk about this particular BCI setup, where a monkey was trained to basically move a point on a screen to some radial dot based on thinking about it, right? So this was recording in Cortex and getting a monkey to learn how to do this. And there have been several interesting empirical results in this setup, which I'm going to talk about and describe, and then I'm going to talk about, you know, can we begin to understand these? What does it look like to understand these from a learning rules perspective? So one of the things that these experimenters found, and I think we find this all over the brain, is that there are these, they talk about them as intrinsic neural manifolds, which is to say, if I've got 1,000 neurons that I'm recording, or say 100 neurons that I'm recording, actually there's sort of like 10 dimensions of real variation there. So that is to say there's lots of correlation. I can run a PCA on a bunch of those neurons, and I can be like, most of the variance is explained by 10 of those 10 dimensions. And that's going to be really crucial for this. What these researchers found in particular was this. Hopefully, can you see my pointer? Yes, awesome. So what they did was they'd go and record a bunch of neurons, 100 neurons. They'd do this sort of factor analysis to say, which are the factors of variation. And then they found, really interestingly, that this in manifold activity, so activity that was really moving around in this lower dimensional intrinsic manifold, it was easier to do learning tasks in that intrinsic manifold, and it was hard to do it for the outside of manifold learning. The way that they did that, and that's one of the really cool things about these BCI setups, is they set this thing up so that they could tease apart what they call an outside manifold perturbation and a within manifold perturbation. And they could basically perturb either within manifold or outside of manifold. And then they could look at the resulting learning effects. So I'll tell you what those look like briefly. Here's one of the schematics from their paper that's probably one of the most extreme versions. So here what they're doing is they've taught the monkey to do this task. Here's success. They turn on the perturbation. In this case, it's within manifold. And you see initially there's a big deviation and the monkey is much worse. And then it quickly gets pretty good at it and hovers around here and continues to improve in terms of how quickly it moves to the targets. In the outside manifold perturbation, they see similarly a degradation, but then the monkey doesn't really improve. It just sort of hangs out here. Similarly, for the time taken to get to the targets, it's not very good. And this is a pretty extreme example that they're showing. This is like, from one of the main figures. But they see this, when they repeat this experiment a whole bunch of times, they see this general trend. So that is that in manifold learning, when you're doing this within the intrinsic manifold that you've figured out, tends to be bigger. The amount of learning tends to be bigger than for out of manifold learning. Okay. I'm gonna tell you just a couple more things, and then get into some of the sort of how we might think about this stuff. I want to mention a related study, which was about this idea of neural reassociation. Okay, so this is a related phenomena in the same BCI setup, in effect. And what they were interested in is when learning occurs. So if I did say the in manifold learning, and I drove a bunch of learning, what happens to the neural representations? So these are supposed to be these sort of like PCA dimensions here, and these are sort of representations, neural representations then in that space. What happens? Was their question, do they update and move far from their original patterns in various ways? Or do they stick really close to the sort of like the patterns from the get-go? And they had they had three hypotheses that they explore, but I'm going to sort of distill these down into two in effect for our purposes here. So they said, here's the neural activity before learning. What happens to it after learning one of these tasks? They talked about two of these things called realignment and rescaling, which involve, I would say, sort of drastic changes of the neural activity vectors that were used to solve the problem. And then they talked about reassociation. And this idea was that you would just sort of reuse the activation patterns that you had already to solve the task. Okay, what they find in particular was this. This was sort of the real data here. And this is repertoire change. So basically, how much did that repertoire change? And this was the prediction from what they called reassociation, which is basically stay pretty close to where you are. These were the predictions from realignment and rescaling. And you can see, in effect, the representations really didn't change much at all. Okay, so I've just told you about two of these phenomena that we find in these BCI studies. What potential candidate explanations are out there? So these two phenomena are what explains this within versus outside manifold learning differences, and what explains this kind of neural reassociation, where actually you don't see big updating of the neural patterns to solve this stuff. And it's worth saying that past work has pointed to a variety of hypotheses, sort of like things like connectivity patterns, corrupted feedback signals, constraints on weight change, sparsity, many of these sorts of things. And there's a recent paper, actually, this Hennig 2021, which summarizes some of the hypothesized explanations out there. What I'm going to talk to you about a bit today is we think perhaps there's a really simple explanation for both of these that just comes out of applying gradient-based learning rules pretty directly. And that's what I'm going to try and walk you through. So in particular, the idea is gradient-based learning, which starts in the current location in parameter space and hill climbs toward better behavior, really generates both, I think, of the essentials of these experimental signatures. There are lots of details in these studies which are not captured, but the essential signatures I'll show you are captured. So here is, for us, and I want you to really have this in your head, the basic in silico BCI adaptation setup that we're working with. This one is really simple compared to the monkey one, but I think you'll see simplicity really lets us dig into this and figure out what's going on more precisely. So what we do is we literally say, hey, here's an image, here's a dot I want you to reach to. This is just a multi-layer feed-forward MLP. The network's going to be taught to push up and output the target position. So it needs to produce this target position and say, I want to move here. Once it's been trained to do that, we're then doing what people do in BCI experiments, in essence, and dropping into a bunch of these neurons, recording from them, and then we're asking it to do a new task based on our computer here. And in particular, we sort of form this BCI projection U, which maps from, say, 1,000 units to the 10 principal components that we've looked at, and then to the two outputs which specify the coordinate to move to. Okay, and because we've set things up like this, we can do in essence the kinds of things that they were doing in the study, where we can look at things that are more in manifold versus things that are more out of manifold. Okay. I'm going to tell you about doing this learning with two very simple learning rules, which I think most of the people in this audience, given the last few talks I've seen, are pretty familiar with. One of them is backprop, and one of them is node perturbation. One of these is simpler, higher variance, slower to learn. The other one, backprop, is faster. I'm not going to dwell on this. That's the gist. So in effect, what we're going to do is learn with both of these algorithms to perform the tasks, and we're going to then learn to perform the perturbations. Just before I get there and show you what the learning looks like when you do that, I'll just note this, that neural manifolds are pretty natural. These sort of intrinsic low-dimensional manifolds are really natural, even in simple neural networks. So if you just instantiate an MLP and push a bunch of activity through it, structured input activity, like those images we saw, you basically get this pattern already, which is to say that a small number of principal components, say 20, captures a lot of the variance in that neural activity. So that happens quite naturally. Once you train these things with backprop and node perturbation, you sort of get even more of that trend. Anyway, something to bear in mind. So you basically see those kinds of intrinsic manifolds form just by training these things. Okay. Here is perhaps the main empirical result to grab onto, which is, in effect, when we move our perturbation from in manifold, which is sort of these low principal components, or the biggest principal components, out further and further to the sort of out of manifold perturbations, principal components that are further and further out. We see this trend clearly both with backprop and with node perturbation, that the things that are more in manifold are faster to learn, slower, and slowest as you go out further and further out of manifold. Okay, I think it's also interesting to note, I don't know how much to make of this yet, but that we see the same pattern in both cases. We see maybe more in the context of node perturbation, that is to say that this one at 7 and 8 is sort of significantly worse than the bigger principal components. We can look at this in a summary figure as they did in some sense. So we can repeat this kind of experiment many, many times, and we can basically sort of look at the spectrum of in manifold versus out of manifold and look at a summary of the amount of learning, and basically this drops off and drops off and drops off. So the essential effect, I'm arguing, we think can be captured just by running sort of the simplest vanilla gradient descent algorithms on simple networks. And you see this basic effect pop out. I'll take one moment, and I'm not going to dive in and talk in detail about proofs. But one of the things that we did in this work, and Peter did some amazing work here, was that we developed an even simpler setup. So forget about a multi-layer MLP. We're talking about like a single linear, a network of a single layer of learnable weights. This is linear. This is based on analyses that Werfel and Sachs introduced to prove things about the speed of node perturbation and backprop for really simple networks. So basically, they were able to prove things about the maximum learning rates under certain conditions when the input components were IID. One of the things that Peter was able to do was he was able to say, okay, what if you have correlated inputs, which is definitely the case in these BCI experiments. In some sense, that's the whole point, right? When I do those analyses, we see tons of correlation. So he was able to extend that analysis and then prove things about the maximum learning rates possible when in this context. And in particular, the maximum learning rate turns out to be determined by the ratio of the variance of the principal components that the BCI is targeting as compared to the total input variance. And I'll try and frame that for you in a moment intuitively. I'm not going to go into the proofs, but the proofs are there, and please go take a look if you're interested. This is what the theory looks like then and predicts. So these are in these simple linear cases. The dashed line here is theory. These are the empirical. If you run a bunch of simulations, you see these things match pretty well. And this is what the particular expression looks like precisely with regards to these variances. Okay. I've told you something about a proof, but honestly, if you're like me, you have to go and sit down with these things. I'm not going to, you're not going to get it in a talk. What I want to do for a moment is try and lead you a bit from the theory to the intuition. And I think that there is then a fundamental intuition we can get to that gets us to understanding, why is it the case that this is so much harder for animals to learn, and indeed so much harder even for these really simple networks to learn. And basically the reasoning just goes like this. A BCI experiment requires a network to produce target activity patterns conditioned on specific inputs, right? So that's what you're asking to do. If the target activity pattern is only a small component of the activity, so it's out of manifold, it'll be harder to pick out the learning signal associated with it out from the variance induced by the other components, and learning will be slower. So in essence, this is just a signal to noise ratio thing. There's a bunch of noise in these components, and if you've said, look, I want you to construct your output based on these out-of-manifold components. They're smaller signals amongst the noise, and you can learn them. You can learn over time to take advantage of them and use them to do the right thing, which is to output the targets that you want. But it will take time, longer amounts of time, to find that signal in the noise. And that's these algorithms can do it. It will just take them longer. Okay. Cool. I think I'm actually doing good for time. I sped up a little to sort of try and accommodate the kerfuffle. I'm now going to talk a little bit about the neural reassociation stuff. So if you'll recall, it was this business about in experiments, when they ran, in this case, let's just say the in-manifold learning stuff, what happens to the neural activities? So basically, when they frame that paper, they say there's a couple different ways that the neural activities could have changed in order to make sure that the task is done well. And they propose these hypotheses, and in fact, when they look at the data, they see that the neural activities update hardly at all. So anyway, we basically went in. This is just the learning curve for the experiment. We had a phase before perturbation where we just trained the network. Then we perturb. And we're going to look at these points, 1, 2, 3, along the course of learning. So what does this look like? It basically looks like this. Here is the cursor. So this is sort of in the real world, if you like. This is what the network is producing. And you see that when it starts, it's pretty good at this. It's basically mapping the things to the right places. Then you apply the perturbative training. I'll come back to that in a moment. At the same time, this is maybe the key thing, I'm plotting along the bottom the PCA components 1 and 2. So I'm basically just like going and doing that PCA analysis, grabbing PCA component 1 and 2. You'll see that these don't map to the same places because there's no reason that they should. The number one thing maybe to take away over the course of this is that as there is massive rearrangement in the space of pointing or reaching, there's almost no rearrangement at all in the PCA space, right? So in fact, by this point, you've got sort of like everything has arrived at where it should be at the end of training for the cursor, and for PCA, almost nothing has updated here. So in particular, if you want to look at it, it's the blue versus the orange here. So that's after gradient descent applied. Now, I'm going to talk about what's being called in this figure a random solution, which is probably not the best name, but I'll try and describe what I mean by that, which is going to give us some intuition about like what the hell is going on here. Why do we see this pattern where sure the real world is changing and not this? So in the random solution, just what we're doing there is we're saying before you start hill climbing, so before we start executing gradient descent and hill climbing on the task, we're first going to just like randomly choose a new initialization for the network, and then I'm going to start training, right? So the green, basically you see here for the cursor ends up in the right place. Like basically it's like, yep, I do the task. Like you just initialize the thing in some random place, then you train it. In the PCA space, it ends up in some completely different spot, right? And I think this is like, then hopefully this is going to begin to sort of get you, get this sort of insight moment that I had when looking at this, which is like, yeah, maybe kind of course. I'm going to dive into this a little more and show you some summary stuff, but I'll try and come back to that intuition. So in effect, I think hill climbing just basically directly predicts association. This is a summary plot where we've gone in after and kind of said what's going on with respect to when I just do my normal hill climbing stuff. This is like before, after, and then the random. And you see the randoms just like reliably has much less overlap with the solutions that were found by starting in a place, doing hill climbing, and getting a decent solution. To kind of frame this back in something a little more like the language of the original empirical work, which was kind of looking at this repertoire change, we can go and do that same analysis on the network, and we can basically say, hey, what happens when you do gradient descent? What kind of repertoire change do you have? Versus what kind of repertoire change do you have if you basically initialize somewhere and then do gradient descent, right? Okay, so why does this make sense? I kind of already hinted at it, but I think we have not developed theory for this because I think in some sense it's almost so blindingly obvious when it's been put like this that you probably don't need theory. Maybe there's something interesting to say about it, but I think this captures it. And that is basically this. If you do if you do hill climbing, in a case where the network has a lot of parametric redundancy, right? So that is, there are lots of ways to achieve its objective, and you're just hill climbing to a good solution from where you are, you will tend to find a pretty good solution for the behavioral task out there without having to move very far from where you are in weight space. And that's basically all this is coming down to. So another way to say that is that the studied BCI tasks that people have looked at in these cases are pretty under-constrained, and the networks that are learning on them are pretty over-parameterized. And that's what's giving us this effect. Okay, so we can kind of test that in a pretty strong way in silico, and maybe people will be interested in going and trying these kinds of things in the real case. So here's a case where we said, okay, look, you don't get to wiggle around. We're going to cut, in some sense, we're going to cut out a bunch of the redundancy that you have. So we're going to basically, when we make our BCI mapping, we're going to put a whole bunch more constraint in, which means that you're forced to change your neural activity to do this. And indeed, when you do that, you see the following, which is, here you are before learning, there's the perturbation, you learn, that's the end of training in cursor space, In PCA space, when we go and look at the neural activity, we can see radical changes in those neural activities. So totally unlike this reassociation that was generally found. And that's because we've basically said, you can't escape it for this behavioral task because of the way that we map from the neural space to the behavioral task. So we expect you would see this in real cases if you went looking for it. Okay, very briefly, I'll just say you can kind of see that in a trended way as well. So I've shown you one example here, but you can show this in sort of a trend as well, where we basically increase the constraint from left to right here, and we're seeing more and more repertoire change induced by that. All right. Now I'm going to do something that I've rarely had a chance to do in a talk, and that is talk about some concurrent work. This is probably one of the coolest sorts of things that can happen in science, and I want to give it some serious time. So we were working on this work. We recently came across some folk, actually here in Montreal at MILA, who are doing a totally concurrent line of work, and I think basically they've discovered essentially the exact same phenomena, and added some new insights to it as well. So I'm going to actually talk about their work. I think that they're around the conference. I have permission to talk about it. We basically were like, just got together and geeked out. Like, we're literally seeing the same things in these experiments. It's kind of, I think, the coolest kind of moment you can hope for in science. So this is Alexandra, Guillaume, and Amy, their collaborator. So what did they do? It looks really very similar. They've got some things. This is actually one of the nicest things about this sort of thing in science. They, of course, haven't done it quite the same way, and they see the same sort of robustness of results, which I guess, to my mind, adds more credence to the fact that we didn't just make something up. So their model looks a little bit different. I'm not going to go into the details of it, but in effect, it's an approximate model of recurrent dynamics. They train a similar network to output, like, hey, move to these targets. That's their loss function. This is an example of the actual network, actually outputting stuff. They also applied their perturbations in a slightly different way. They're in and out of manifold perturbations. And actually, nicely, I would say, they apply their perturbations in a way that's more true to the original empirical work than we did. We chose ours for sort of analytic convenience. They actually did it like truer to the original paper. So they used these perturbation matrices, PWM and POM, to either do a perturbation on the within manifold or out of manifold, or out of manifold. And these are the kinds of perturbations that they got. That's just what it looks like once they've done the perturbation. And here's their main result, and it's just dead on. So that is to say, they're like, what happens with the loss? Very clearly, the out of manifold learning is slower, just slower for these things. Okay. Not only did they capture the main result, they had this figure in their recent manuscript, which I really want to dwell on for a moment, and I just think it's like just lovely. It captures a couple things which we haven't really talked about in our work. I think it sort of gives us a bunch more insight about some of the really cool dynamics that researchers see. So they identified that adaptation dynamics depend on learning rate. And I'll just describe what's in these two figures a bit, and then maybe go into a bit of interpretation. So here they're basically saying, I'm going to set the learning rate to something smaller. And there you see a difference. So that is, again, you see that the within manifold is faster than the out of manifold. And you see a lot of variability there for this learning rate. In this case, and this one's the kind of amazing one, when they've set this learning rate a bit higher, you see everything from sort of not doing as well to ringing to completely flatlining and being unable to make progress on the objective, which actually you see it looks like in monkeys. So often what will happen initially is those monkeys will really not make progress on these tasks. They'll just be kind of dead in the water. And I think that these kinds of plots give us a bit of insight into what's going on here. I don't know if you can see, but this is the empirical figure where you saw a variability, like sort of a variable amount of learning as a consequence of the in manifold versus out of manifold. And sometimes that includes even zero learning. Finally, I think just rounding out on some of the new things they did, here is a specific example of a case where they found an out of manifold perturbation where the network just couldn't learn it at that learning rate at all. So it basically just flatlined. And this is particularly interesting, I'll say for a moment, what the sort of next piece of this, because they're interested in downstream clinical stuff. And one of the things you might really want to do is if you've got a case where you've hooked up some sort of BCI component for someone, if they can't initially learn it, are there any tactics that you can use to get them to have a better go at learning it? And what they've shown here, as people have explored a bit in the monkey case, is that you can do these incremental adjustments. So what they do is they basically say, here's the out of manifold hard problem we're trying to learn. Here's some sort of in manifold thing. Can we walk along from the in manifold to the out of manifold perturbation that we're really interested in? Can we walk along in a series of stages and learn that? And indeed they do. So they solve initially really easy problems, increasingly hard, but importantly, this point that they get to is much, much better than the point they got to here. Anyway, so the work contains some really cool stuff. I'll try and have a pointer to it in a moment. I have just 2 more slides. I have tried to pick up the pace a fair bit. We're doing good. Okay. I just wanted to mention very briefly that, 2 more things. We've done this in as simple models as possible, largely. Recently, though, we've kind of gone and done some of this work in the context of dynamic recurrent networks, where we actually get networks to generate reaches, and we see these kinds of differences again when we introduce perturbations. And finally, I want to say, we think we've also found the basic phenomena in a much earlier work, in a really nice paper from Fulner and Klopath in 2020. So in one of their supplementary figures, they actually have an example of doing some of this stuff where it looks like you can see a difference between outside manifold and within manifold learning that's quicker. I haven't really dug into it with them, but I wanted to mention it. I think that maybe there was some of this was in the literature already, and it would be cool to go and think about. I'm going to round out in summary and just say, okay, we've explored these 2 notable BCI phenomena, the within versus outside manifold learning differences and reassociation of neural representations. I think we have some evidence now that both phenomena may just be straightforward consequences of gradient-based optimization processes, node perturbation or backprop, for example. Theory and intuition can help make sense of these phenomena. The thing that I'm maybe most interested in is being able to use empirical results and to wiggle back and forth and get a bit better and increasingly good idea about what kinds of learning algorithms we think the brain is using. Super nice. We have this concurrent work which seems to corroborate these experiments. I think Guillaume is around and Alexandra. Find them if you're interested in this stuff. And I'm really hopeful the BCI experiments might help us elucidate learning algorithms in the brain, as that's my kind of hobby. There's a preprint. If you're interested, take a picture up on bioRxiv. I also want to thank Alexandra and Guillaume for a bunch of productive discussions. They have this concurrent manuscript available, I think, literally in the next day or so, up at this address on bioRxiv. And that's it. Thank you. Go for it.
Can you point if there is any relation between the reassociation you're talking about and lazy and rich learning? Is that just that you set up the network to be in a lazy format, therefore you end up with reassociation?
There's a bit of noise. I caught part of that. Is there could you repeat?
Yes.
Yeah.
Is there any relation between the reassociation part of your talk and the very nice work on lazy and rich regimes for learning? And does it so happen that your networks and the setup is just in a lazy regime and therefore what you're seeing is mainly reassociation?
I'm extremely sorry. I caught about half of that. Association. Yeah. Okay. Yeah. Is there a relationship between the two sort of phenomena? Potentially, the two.
Rich and lazy learning and reassociation. Do you just end up being in a lazy learning regime and therefore you're going to just do reassociation? If you were to change the setup or to do it in such a way that you're going to be in a rich learning regime, you could end up with very different mechanisms to point towards.
Yeah, okay, I think I've got you. So in effect, is there, are we seeing, okay, I think if you had other kinds of cost functions, and maybe this is what you're getting at, if you had other kinds of cost functions, there are probably things that you could do to induce, radical changes in the neural activity patterns, even in this context. So I think that is true. I think so. There is a sense in which then there is an implicit assumption, which is that we're basically saying, hey, and this is what we're doing in our case. We're basically saying, just solve the behavioral task. I don't really care about anything else. And just aim for that and just do it with some sort of a simple gradient-based estimate and update. Does that make sense? Yeah, okay, sorry, grab me after, I may have, yeah.
Hello, hey.
Hey.
Thanks for the nice talk. So I have a kind of a high-level question. I'm wondering, so you're focusing on a relatively particular learning problem where learning is localized to a small circuit in the brain. So I'm just wondering, to what extent do you think the results would inform other types of learning problems, say across the whole brain, or maybe more extreme learning, say during development where the whole circuit has to be shaped? Do you think these types of insights translate to those cases?
I'm going to be honest and say it's a good question. I haven't thought about it much, and so I'm basically at this point going to be like, I'd be sort of really stretching. My guess is to be honest that there's going to be elements of learning across the brain which you could get with these kinds of things, but there is inevitably, I've tried to paint a simple picture, but I think that there's a bunch of complexity in actuality there that isn't going to be captured by these kinds of things in at first in first go. So even in the context of the BCI stuff that I talked about here, there are some bits of the phenomena which I don't think we see really naturally in these very, very dead simple experiments. So I'll just mention one of them. In the context where the monkey does the out of manifold perturbations, the learning really looks like this, or rather, when the perturbation is induced, they really get worse immediately, and then it's pretty dead. It's pretty like straight across, and then when the perturbation is released, it's bam, they just snap back because in effect they've learned nothing. And those kinds of phenomena I don't think are captured by this sort of first pass sorts of simplifications we're making. Yeah. So probably no is the generic answer.
One last question.
Do you think using the techniques you talked about in principle, you could tell the difference between different variants of gradient descent or something like that?
Lovely question. When we set out on looking at this, I should say that in some sense, the motivation to go down this route of picking it at BCI was to do exactly the kind of, aim at exactly the kind of question that you just asked, which is if we look at these BCI phenomena, can we go back and make better inferences about the underlying learning algorithms? In a sense, I think that we've have a negative result there, ish. So, and by that I mean, like, we kind of have seen that both for node perturbation and for backpropagation, we see roughly the same phenomena. Now, like, the precise timescales and so on, I don't think are matched, perfectly to the empirical stuff, and maybe one of them would be matched better, but I'm not sure if I believe the details down to that level. So that's precisely the direction we'd hope to go in. I'm not sure if we have a handle on that. I will say that in the Fulner and Klopath paper I mentioned, they have some experiments where they actually don't see a difference between in and out of manifold learning in some conditions. And in particular, it looks like, so in particular, in those experiments where they don't see a difference, they are running RLS, this like recurrent least squares algorithm, which is way more powerful than backprop, than just like an SGD. And in that context, they don't see it. And I think that may be because that algorithm is doing like an online learning rate adjustment schedule. So that is, maybe there is a bit of some inferences you can make, but I've kind of regarded it up to this point as a bit of a negative result in this front, even though that's precisely what we had hoped for from the beginning. Yeah.
Okay, let's thank all of the speakers of this session and for hanging in until the very end.
All right, I get to be in charge of the closing remarks. And so on behalf of the executive board of Cosyne, I want to extend an incredible and huge and very loud thank you to Jess and Blake for organizing a fantastic program. And I also want to thank Laura and Tim, our general chairs, who provided all the excellent support and worked out all the travel grants and all the awards that they did. There are countless people to thank, but one of the most important people is Leslie Weeks, who's actually not even here, but so we have to clap really loud. She's already at Tremblant. And everybody else who worked so hard to put this meeting on and make it such a huge success. I really want to thank them. I want to thank all of the session chairs. I want to thank all of the speakers. I want to thank all of you for coming and making this such an amazing meeting. So much to learn, so much to see, so much to do. So thank you all for coming, and please come see us at Lisbon in 2024. All right, off to the buses.